Hello, my name is Johanny Guzman, and my professors' names are Lisa Friend and Sarah Rogers. Today, I will begin by discussing with you the differences between the two cell walls, the gram-positive cell wall and gram-negative cell wall. So the first and most easy to identify difference among the two is the peptidoglycan layer. So the peptidoglycan layer is made up of sugars and amino acids and provides structure and protection to the bacteria. So then in gram-positive cell wall, the peptidoglycan layer is a thick layer, while in gram-negative, it is a thin layer. My next difference among the two is that gram-negative cell walls contain an outer membrane and a periplasmic space that the gram-positive cell wall does not contain. This outer membrane contains lipopolysaccharides, or LPS. The lipopolysaccharides provide the bacteria with protection from harmful environments and harmful substances. This trait is what makes them more resistant to antibiotics and as so harder to treat when found in a patient. If they are successful in entering the body, they can be seen as symptoms of fever, inflammation, and blood clotting. My next difference is that gram-positive cell walls contain teichoic acids or lipoteichoic acids that the gram-negative cell wall does not. These lipoteichoic acids or teichoic acids provide a more stable or stronger structure in the cell wall. My last difference among the two is the color they stain. So the gram-positive cell wall stains purple, while the gram-negative cell wall stains pink. So then my next topic I will be discussing is the procedure of completing a gram stain. So you first begin by making your smear or preparing your smear. So you do so by dropping a drop of water on the slide, of clean sterile water on the slide, then sterilizing your loop and picking up a single colony and smearing it thinly on the slide. Then you sterilize your loop and you can place it on the side. Then you let that air dry. And then you heat fix it by placing it on the Bunsen burner just for a couple seconds, not too long. And then you place it on the slide tray and you begin your staining process. So the first chemical that you will drop in is crystal violet. This is a purple dye, and it's known as a primary stain. This will stain the whole slide, so the whole thing. You let that sit for a minute, then you rinse it out. The next one is the mordant, which is iodine. This mordant, it serves the purpose of fixing the crystal violet. It will help the crystal violet be non-soluble or not dissolvable, and that's its purpose, basically, just fixating the dye. So then you let this sit for a minute as well, and then you rinse it out with water. Then comes in your decolorizer, which will decolorize the gram-negative cell wall from the crystal violet. The decolorizer is ethyl alcohol. This one you put a couple droplets in, but you only let sit for 5 to 10 seconds or as provided by the professor. You remove immediately with water. So 5 to 10 seconds, not more, or you will ruin your results. You will not get accurate results. And then you rinse it with water. So then in comes your counter stain, which is safranin. This will stain the gram-negative cell wall pink. And then you let that sit for a minute, and then you rinse it out with water. You then can essentially, you blot dry the slide with bibulous paper, and then from there you can view it under a microscope with immersion oil to get a clearer view. So then takes me to my last topic, is telling you how it helps medical professionals. Well, this provides us with the type of bacteria that it is, since it's either gram-positive or gram-negative. It provides us the size, which we can see because it's stained a certain color, the size, and the shape. These three factors help us narrow down the type of bacteria that can be causing the infection or provoking the infection in the patient. And essentially, you might undergo other processes to really narrow it down and figure out which one it is, and then finally find out what is the cause of infection and so treat the patient. And that's all I have. Thank you.